All right, Unit 5, we are in Chapter 10, and we are starting to learn about the nervous system. Now, Chapter 10 is going to be the microscopic neurons and the neuroglia that support it and how they work and what is an action potential. And then Chapter 11 is about the larger structures like the brain and the spinal cord and how those work. So, just an overview of the nervous system. It is the master regulatory system. I mean, it controls pretty much everything else. It sends and receives information. It takes in what's called sensory input from the environment, whether that be internal or external. It integrates and processes to determine what to do with that information that was just brought in. And then it can respond by motor output, which will talk to muscles to move, like we saw in muscle contraction in our muscle unit, or it might talk to glands to respond. Like it could be salivary glands, or it could be an endocrine gland. And it helps maintain homeostasis. It really keeps you alive, and it is the center of thought, learning, and memory. The main cells of the nervous system, as we learned in our tissue unit way back when, are the neurons, the nerve cells, and they are what conduct these electrical impulses using the neurotransmitters. Remember acetylcholine and how it was released by the neuron to talk to a muscle? That's what they're talking about there. Acetylcholine's not the only neurotransmitter. In fact, I'm going to share with you some others. But there's also neuroglia. And remember, the glial cells are what help protect and support these neurons and make them able to do their job even better. They do not conduct electrical impulses like the neurons. And they're also much smaller, if you'll remember from the tissue unit. And so the flow of information, we receive some type of sensory receptor information. So it could be a stretch receptor internally. It could be, hormone changes, or it could be blood, a component in the blood is too high, some type of receptor, or it can be an external receptor like a optical receptor. This is actually what we'll study in our very last unit, Unit 6. And it brings in information and sends that to the brain and the spinal cord. We'll call that the central nervous system. So this is peripheral nervous system. I would write PNS right here, peripheral nervous system. And then right here, the brain and the spinal cord is CNS, the central nervous system. And the brain and spinal cord take in the sensory information and kind of decide where this information needs to continue and what the response should be. A very complicated process, and then it sends it to the peripheral nervous system again, where we call this motor output, and it talks to the effector cells. What cells are going to be affected by this sensory input? And so, like we mentioned, it will be muscles and glands usually. And this is also part of the peripheral nervous system, because it's going off of the sides. So these are the sides, and this is central. The sensory receptor is what is found on what's called sensory neuron, and then the effector will be affected by whatever the motor neuron transmitter is. So as I mentioned, the central nervous system composed of the brain and the spinal cord, where your peripheral nervous system connects to the central nervous system and all the other parts. So these nerves, cranial nerves and spinal nerves coming off of those areas, integrating different parts of the body. And there are two subdivisions. They're the afferent, afferent meaning coming into the brain or coming into a particular place. It happens to be the brain here, so that will always be a sensory neuron. And then the efferent, exiting the brain, will be a motor neuron. So let's talk about the sensory or afferent division first. So this is where sensory information is detected, or there's changes detected, and the receptors start to make an impulse that runs down this nerve heading to the central nervous system for integration. The motor division will take a transmitted impulse from the central nervous system to the effector cells, whether it be muscle or glands. And there are two subdivisions of the motor division. And these show up so much later on in anatomy too. Somatic Soma means body. And so somatic transmits your voluntary skeletal muscle. We just studied that, how skeletal muscle is controlled by you somewhat or most of the time. And so they call that you're moving your body, somatic. Autonomic sounds like automatic, and so it's going to transmit the involuntary commands. So it could be smooth muscle or these glands they mentioned. And so here you can see the brain and the spinal cord is the central nervous system, and then you have these 12 cranial nerves coming off of the brain, and then you have tons of spinal nerves coming off of the spinal cord, and this is help controlling and bringing in information. So it's going to be sensory information coming in, and sometimes the same nerve can do motor information coming back out. Pretty cool. So again, we have some sensory receptor that tells this afferent pathway to run down a sensory neuron to somewhere in the central nervous system where it's integrated. And then information is an action potential actually is generated and moves down a motor neuron either through to skeletal muscle or to the autonomic nervous system. So I've got quite a few students and friends that experience migraines, about 12% of the population they estimate, and this is a pounding head, often nausea, shimmery images. My stepson had migraines as in his teenage years, and sometimes the environment can trigger this, like bright lights or lack of sleep or stress, too much caffeine or alcohol, and hormones can trigger this in women. Oftentimes they'll see women who experience a lot of migraines will have those just prior to menstruation. And this migraine can last anywhere from just a few hours to days. There is a period of excitation followed by the unresponsiveness in particular neurons that stimulate the production of these pain sensations. And the good first approach is to identify what triggers this migraine and then avoid those. So there are drugs that help stop migraine attacks. In fact, what they do is constrict some of those blood vessels. Botox has shown that if you get injections of that, they help reduce migraines. But everybody's different, so it's what therapy works best for you. All right, so let's talk about neurons, the main cell of the nervous system. They vary in size and shape. They vary in the length and the number and size of their axons and the dendrites. They can be really crazy looking. We'll look at some. But all of them, no matter what size, shape, where they're at, how many axons they have 3 major parts. The soma, remember, somatic means body. Soma is the cell body. So this is where you're going to find the nucleus and the majority of the cytoplasm, a lot of the organelles and stuff. The dendrites are branched receptive surfaces, and a neuron may have tons of these. And so this is to receive information from different places. And then the axon is what transmits the impulse and eventually releases the neurotransmitters to either another neuron that it's talking to or the effector, which could be a muscle cell or the gland. A neuron can only have one axon, but it can have many dendrites. And so axons are the branches that receive information, and they do lead to an axon hillock. This is an area where the cell body starts to arise from these axons. There's collaterals or branches from axons, and the axon, sorry, I just misspoke. Let me correct myself. The axon hillock is where the soma dips down and becomes the axon. Collaterals are branches from the axon. The axon terminal, we've used that term before with our muscle unit because that's these extensions from the axon that we're going to find the synaptic knobs, these rounded endings in the synaptic terminals, and that's where we have those neurotransmitters. Schwann cells are a type of neuroglia only in the peripheral nervous system, so only in sensory neurons and motor neurons. And what Schwann cells do is they wrap around, kind of like a tortilla, they wrap around the axon, and they're very fatty, and they create what's called myelin, which is a mixture of this fat and protein that fills these layers by the Schwann cell membrane. And what that myelin sheath does is insulates the axon so that the transmission of the signal runs faster. Think about the axon like a wire, and the myelin sheath is the protective plastic covering on the outside that makes sure that the electricity runs in one direction, it runs quickly, and it doesn't divert and go into your body. The nodes of Ranvier, I think he was French, Ranvier. If you're, from the South, you can say Ranvier. But these are going to be gaps in the myelin sheath between the Schwann cells. And so let's look at this. Here's the soma. You see some organelles and the nucleus, a very identifiable organelle. And so this would be the soma or cell body. And then these are the dendrites coming off. So this one has 1, 2, 3, 4, 5, 6, 7 dendrites. And sometimes those dendrites have branches that come off of them. And no big deal. All right. So here's your axon hillock where the soma, it looks like a hill, like you could slide down there, going to this axon. And so don't worry about neurofibrils, but here's this axon, and the axon runs very long, and they can branch this axon, right? We mentioned that. And these are the Schwann cells. So 1, 2, 3, 4, 5, 6, 7, 8 Schwann cells in this picture, and together they make up the myelin sheath. And the myelin is all this fat and protein that the cell membrane, remember the cell membrane is mainly made of lipids and proteins, that insulates this wire, this axon. The nodes of Ranvier are these spaces in the myelin sheath, and that's going to help us with what's called saltatory conduction. When you get down to the axon terminal, you'll see that synaptic knob, and we zoomed way in on this in the last unit where the neurotransmitters will be found. So these can all talk to different effector cells, whether it be another neuron, a muscle, or a gland. And so this is showing a really good picture of the myelin that insulates these axons. Now, these are called Schwann cells because they're in the peripheral nervous system. If these neurons were in the central nervous system, they named those cells something else. They're actually one of my favorite words, oligodendrocytes. But these are, because they're Schwann cells, they are located in peripheral nervous system. And so you can see that the axon here, and these layers of myelin sheath. And then they call this fatty extra outside part the neurolema, where a lot of the organelles have been squished to. Remember, this is one cell that's shaped like a tortilla that's been wrapped a lot around this wire to insulate it. Okay. So not all axons are myelinated. So myelinated axons are coated with the myelin sheath, and they're produced by Schwann cells if they're in the peripheral nervous system. Here's my favorite word. They're produced by oligodendrocytes if you're in the central nervous system. When you see groups of myelinated axons in your central nervous system, whether it be it's in the brain or spinal cord, it's oligodendrocytes, right? Because oligodendrocytes are what causes the myelination in the central nervous system. But that's what white matter is. So maybe you've heard of white matter in the brain. All that white matter is myelinated axons. And what does myelination do? It increases the speed for these impulses. Now, unmyelinated axons are encased by Schwann cell cytoplasm in the peripheral nervous system, but there is no wrapped coating of myelin. And so groups of unmyelinated axons are in the central nervous system are called gray matter. So we're going to have white matter and gray matter. So here's the difference. You see a myelinated and an unmyelinated axon. Multiple sclerosis, star this. I think this is on your test. This is where the myelin sheath is destroyed by your own antibodies. The antibodies tag the myelin sheath as foreign, and your body starts to fight them, and it leaves these scars. And that stops the neuron from conducting the impulses. So without input from motor neurons, muscles stop contracting. And remember, if you don't use your muscle, if you don't use it, you lose it. So it's going to really affect movement, and it can affect mood and vision and all of the things. There are treatments involved, but they suppress the immune system so that they'll stop attacking the myelin, so then you could get a secondary infection. I do have a relative that has multiple sclerosis, or MS. So let's classify some neurons based on their structure. We already have kind of classified them on what they do. We talked about the afferent pathway having a sensory neuron, the efferent pathway having a motor neuron, and then the neuron in the central nervous system is going to be called an interneuron because it's between the two others. But let's look at how they're structured. There are multipolar neurons, and that just means you have a lot of extensions from the cell body. You could have many dendrites, but always remember just one axon. If you have and this is most neurons. Bipolar neurons only have two processes, so you're only going to have one dendrite instead of many, and one axon. Always one axon, right? We see these in the ears, the eyes, and the nose. So not that common. Sensory. And then there's unipolar. We don't use pseudo-unipolar, even though it kind of makes sense, but unipolar, just one process comes off the cell body. And what happens is that process then branches into an axon and what am I trying to say? Dendrite. Let's look at a picture of them. So here is your multipolar. So off the cell body, 1, 2, 3, 4, 5, 6 extensions coming off there. Bipolar, just 1, 2 extensions. So one dendrite, one axon. And then here is the unipolar. It's just one extension and then it branches. Now let's classify them by function, and we already mentioned this. The sensory or afferent neurons carry impulses from the outside to the brain. Some are unipolar and some are bipolar, or most are unipolar, Interneurons, that's going to be the ones in the central nervous system, and they're multipolar. And then motor neurons, which are in the efferent pathway, carry information from the central nervous system to the effectors, and they're usually multipolar. Now, in some somatic, remember that's going to be in somatic nervous system, those motor neurons are going to skeletal muscle, and in autonomic, they are going to the other effector cells. And so I love this picture. It really helps you see the flow of information coming from the outside and how you respond to it. Now let's look at the neuroglia that support these neurons. And in embryos, they guide the neurons into position, and they may even kind of wake them up and specialize them. They do produce growth factors to nourish the neurons, and they remove excess ions and neurotransmitters. So they're kind of like the babysitters of the neurons. There are astrocytes. Astro means star, so they kind of look like a star. They connect neurons to blood vessels and help exchange nutrients and growth factors. They can form scar tissue. They regulate ion concentration, and they are part of the blood-brain barrier. I think I've mentioned that before, where the brain is very particular on what can pass through. Oligodendrocytes, we already mentioned, they myelinate the central nervous system and provide structural support. Microglia are little phagocytic cells that also can provide structural support, but they're kind of eating up debris. And then ependymal cells, they line the central canal of the spinal cord and the ventricles of the brain. They help regulate what the cerebral spinal fluid is made of. And they are ciliated cuboidal or columnar cells, these ependymal cells. So here they are. So you can see simple columnar. This is the fluid filled, like spinal fluid and brain fluid that helps protect and feed and nourish the brain. And then here's outside of that. And then what you see here is a little microglia. And remember that it is a phagocytic cell, so it's going to eat up any debris. Look what the astrocyte's doing. It's taking nutrients from blood in the capillary and giving it to the neuron. And so here's this neuron, and it is wrapped in myelin. We're in the central nervous system, so those are oligodendrocytes. Here's another picture of satellite cells. We haven't talked about them yet. And Schwann cells. Notice these both start with S. These are not in the central nervous system. They are in the peripheral nervous system. So the S words are only found in the periphery. So here are the PNS cells, what we just mentioned. Schwann cells, which do pretty much what the oligodendrocytes do. They form the myelin to speed up the nerve impulses. Satellite cells are clusters around the cell body of the neuron, and they nourish the neuron. So here are the types of neuroglia. Notice the PNS has the S cells, and everything else is in the central nervous system. Astrocytes, they're star-shaped, and that's how they branch out to the blood and give the nutrients from the blood to the neurons. Oligodendrocytes wrap around. They insulate the inner neurons. The microglia are the small cells that are phagocytic, part of your immune response. They can also eat up any viral or infected cells there. And then our ependymal cells are those simple columnar, simple cuboidal cells that form this barrier and make the interstitial fluid, the brain and spinal cord fluid, or cerebrospinal fluid. So mature neurons don't divide. That's why if you damage nerve cells, you can't fix them. If the cell body is injured, the neuron usually just dies. And so you can't make another one. Now, there is some neuron regeneration in the peripheral nervous system and in the central nervous system, but you are not going to be held accountable for this information for the test. But this is just showing the site of injury and how it can fix itself. Of course, if you injure this, it's going to die, not regenerate. Okay, let's just keep going. The cell membrane potential. So this is the hardest thing I think you'll have to learn in Anatomy 1, is this idea of polarity on a membrane and how an action potential works. And in my class, when we're in person, we usually act this out, and my students really get a good understanding for this. So I'll try to explain it as best I can virally. So this cell membranes are electrically charged, and we call that polarized. And the reason they named it polarized is because one side has a positive charge and one side has a negative charge. It is potential energy, just like a battery. So having this charge difference is called polarity. So inside the membrane is negative, and so the outside is positive. And this is because of this uneven distribution of ions on either side of the membrane. And this has to happen for an impulse to work in both neurons and muscles. These cells are what's called excitable and can rapidly change their charges. And this charge, this wave charge change, it's going to be a wave of charge change, is called an action potential, and it's the electrical energy that is used to communicate between cells. And so the charge inside the cell is actually measurable, and it's called the membrane potential because there is potential to transport charges across the membrane. It's potential energy. And the resting membrane potential is what value that is inside the cell when it's at rest. Now, don't get me wrong. When a neuron is at rest, that doesn't mean it's not doing anything. It's still doing stuff, but it's not conducting an impulse, okay? So the charge on a resting membrane, right inside the membrane of this neuron, we're like on an axon, is about negative 7 millivolts. Now, some people, it's a little bit above or a little bit below. You've probably heard of people having higher thresholds for pain or whatever. This is kind of what they're talking about. So those potentials, this action potential, this membrane potential, results from this unequal ion distribution, mainly because of the sodium-potassium pump. And we talked about the sodium-potassium pump way in the first unit, and I said, you're going to need to know this later. The sodium-potassium pump, if you'll remember, and you can write this out here if you want, pumps 2 potassium in and 3 sodiums out over and over again. And it is a pump, so it requires ATP. So remember when I said it's at rest, it's still doing stuff, the sodium-potassium pump is still working. And because you're pumping 2 in and 3 out, they're both positive charges. Notice that. They're both cations. But because you're pumping 3 of these out and 2 in, and then 3 out and 2 in, you can see how outside you're going to accumulate a positive charge. And if the outside is more positive, that means the inside is more negative. How negative? About negative 70 millivolts. The negativity inside the cell is due to these large numbers of these ions being pumped in, as well as the proteins that are stuck in the cell that sometimes have a negative charge. This all has to happen for the neuron to be excitable, to work. And these gated channels, remember channels allow for facilitated diffusion, are going to allow the movements of these ions at certain times and cause this action potential, this energy wave of communication to happen. Whew. So sodium and potassium ions follow the rules of diffusion, moving high concentration to low through those channels. And the resting membrane is more permeable to potassium than sodium. If some potassium diffuses out, well, remember outside's positive anyway, so it just further helps the positivity outside. And remember, the sodium-potassium pump is the pump that maintains this balance, pumping 3 sodiums out and 2 potassiums in, and it uses ATP to do that. And it's pumping them in opposite directions against their concentration gradients. So an action potential is a sequence, picture you're at a football field and people do the wave. It's a sequence, like a flow, like a wave of electrical events in an excitable cell. And what's happening is the membrane potential is changing from negative inside to positive and then negative again, and it returns back to resting potential. And so this is showing channels closed, and this is showing ion channels open, and we are able to control the flow of these ions to make the internal environment of the cell negative, positive, and then negative. So here's just a fake neuron before the membrane potential is established. Potassium ions can still diffuse out because remember, they're more permeable than sodium can diffuse in. So you're going to have more positivity out here and negativity all along the inside. So it's negative charge all along the membrane on the inside and positive charge all along the outside, and it's at rest. The membrane potential, negative on the inside of the membrane, aids the sodium diffusion into the cell and opposes potassium diffusion out of the cell. As a result, slightly more sodiums enter the cell than potassiums leave. However, the sodium-potassium pump balances these movements, maintaining that negative inside and that positive on the outside. It's still at rest. If we measured this, it would be negative 70 millivolts. So here we are at rest, negative 70, here we are at rest. So an action potential is where we're measuring one piece of that membrane. Like if we're measuring this membrane and it's negative 70. When an action potential begins, this section right here is going to go from negative to positive and back to negative. And what we're going to see is that's going to start a wave. Once this becomes positive, it triggers this part to become positive. And while this one's become negative, then this one. And it's going to be this wave of changing electrical charge along the membrane. And so if you're going to graph this, star this, you need to know it. Right here is at rest. What is causing resting potential? The sodium-potassium pump. Pumping 3 sodium out and 2 potassium in. And then if you reach a certain amount, it's called threshold. And once you reach threshold, you open all your gates and sodium rushes from the outside into the cell and makes you positive charge very quickly. Look at milliseconds. Whew. And this is called depolarization. So on this side of the graph, like here I would put NAK pump. Here I would put sodium rushes in. Just as quickly, we close our sodium gates and open our potassium gates, and potassium rushes out. So potassium moves out. And we repolarize our membrane. Sometimes we repolarize it too much, called hyperpolarization, but the sodium-potassium pump establishes the resting potential again. So neurons are excitable cells. They can respond to stimuli. A stimulus is anything that can change that resting potential of negative 70 in either direction. So it can make it more negative or towards the positive. When neurons detect stimuli, they respond by changing their resting potential. An excitatory stimulus opens the sodium channels and the sodium enters. The local potential change in the membrane potential, is graded, and once it reaches threshold, it's not graded. It's all or nothing. And so what we see here is there's some stimulus happening here that's causing sodium to come in. And notice we're getting less negative. But it wasn't enough stimulus for us to meet that threshold point. Now imagine if this is saying there's pain and your threshold's right here. You didn't feel pain. Maybe you just felt some pressure or whatever, but you didn't feel pain. And so you didn't tell your brain, hey, there's pain. But if you have enough stimulus that causes you to become less negative and reach threshold, all those gates are going to open, and you're not going to be just less negative, you're going to be positive, and the action potential starts. It's all or nothing, and it starts traveling. You can see the sodium-potassium trigger zones right here around the axon hillock. It'll travel down the axon and release its own neurotransmitters to say, hey, this hurts. And what should you do? How should you jerk back? Should you scream? That kind of thing. So that threshold stimulus is the excitatory stimulus that causes enough sodium ions to flow in to reach that threshold potential, which is usually about negative 55. Now, if yours is higher, it's going to take more stimulus to get you to reach there and feel the pain. If you have a low threshold for pain, it takes less stimulus for you to feel pain. So many voltage-gated sodium channels will open at that trigger zone, and that starts this action potential where you depolarize the membrane. It's all or none. You're either going to reach an action potential or not, depending on if you hit that threshold. And then once you depolarize, you'll just as fast repolarize, maybe hyperpolarize, and return to rest. And so this is a great picture of what's happening. This is at rest. We've got a lot of sodium out and a lot of potassium in. But once there's enough stimulus, it opens up all the sodium channels in this region. And the positivity rushes in, which is going to cause all the sodium in the next section to open. While this section is opening and sodium's rushing into this part of the axon, this section is now closing the sodium channels and opening the potassium. Potassium rushes out and returns that positive outside, negative inside thing. So this is the repolarization period. And then it will go back to rest. Meanwhile, this is all happening all the way down like a wave. So my students and I stand in a line and we act this out, and the students can really see this wave of energy moving down. This is a great table for you to look at and read through to understand how this works. During an impulse, the portion of the axon that's actively conducting the action potential isn't able to respond to another stimulus because it's in this moment of reacting. It's like you can't scream again if you're already screaming. So there is a refractory period, and it has two parts, the absolute and the relative refractory period. The absolute refractory period is this time when that threshold stimulus can't generate another action potential. The channels are all briefly unresponsive because they're recovering, if you will. The relative refractory period is a time when only high intensity stimulus can generate another action potential. Repolarization is not all the way complete, and so the membrane is reestablishing resting period. And so the refractory period does limit how many action potentials can be generated per second. So the speed of this impulse varies with the myelination. Remember, unmyelinated axons conduct the impulse over the entire length because there's no nodes of Ranvier. It's unmyelinated. Myelin is rich in lipids and prevents water and all these ions from crossing. So it acts like an electrical insulator, and the ions can only cross through these gaps of myelin sheath. And so since they don't have to go through the whole membrane and they just jump from node to node, they are much faster. And this is called saltatory conduction. Salt probably because it is sodium and potassium. The axon diameter can also affect the speed. The thicker the axon, the faster it transmits, kind of like a water hose. If you have a thick hose, it can transmit a lot more water. And so here is showing you a thick myelinated axon, how fast it can move this action potential down the axon, and a thin unmyelinated is much slower. And so this is what that action potential looks like and how the conduction moves down the axon. And so this is saltatory conduction. So the action potential started here, but because this doesn't allow electrons to move, they quickly diffuse down here and the exchange happens here. And so it looks like the action potential is jumping. And that's called saltatory conduction. So what factors affect impulse conduction? The increase of potassium, the decrease in potassium, if you're allowing potassium to move across, and drugs can do this to you as well as like diseases or whatever. Like if you're born without as many channels as you are supposed to, the impulse may not reach your brain and you won't be able to perceive, touch, or pain or something like that. Okay, so now that we know how an action potential works, we got to talk about what happens at the synapse. And the good news is, if you learned action potential, synapse is going to be real easy because you've seen it before. The synapse, remember, is a site where a neuron is talking to another neuron or another cell. The presynaptic neuron is sending the impulse to another neuron. The postsynaptic neuron is the one getting it. So it's the synaptic cleft is between the presynaptic and the postsynaptic. So the synaptic transmission is where the presynaptic neuron sends an impulse, and it uses neurotransmitters to do that. And so this should look really familiar. This is how the motor neuron talked to the muscle. Well, this could be a sensory neuron talking to an interneuron. And so here's that synaptic cleft, and the action potential, the wave of energy comes down. It's going to cause calcium to come in. These vesicles are going to release their load of neurotransmitter that will diffuse across this very tiny space and bind with receptors. And if that's enough information, it will cause another action potential to occur. This is a good schematic of it. So here's your presynaptic neuron and your postsynaptic neuron, and it's talking through the synapse. It's always in one direction. And so here is this presynaptic neuron. Here's the calcium coming in, causing these vesicles to release the neurotransmitter. If this is acetylcholine, then these are the receptors on the muscle that cause the action potential to generate there in the sarcolemma. But this, I think, is another neuron in this picture. So transmission of a nerve impulse does use neurotransmitters that cross the synaptic cleft, and they're going to bind to specific receptors on the membrane of that postsynaptic neuron. And every neurotransmitter affects the neuron differently. Some open ion channels, some close, some do other things. But there are two major types of neurotransmitters: excitatory, which increase the permeability of sodon, which increase the likelihood of generating an action potential, and inhibitory neurons or neurotransmitters, and they actually make the neuron polarity inside more negative, away from threshold. So, for example, like a drug, heroin, I don't know, morphine is an inhibitory. So it hyperpolarizes your membrane, and so even if you have pain, it's never going to reach threshold because it's causing you to be more negative inside your neurons. So there's excitatory postsynaptic potentials and inhibitory postsynaptic potentials, EPSP and IPSP. And this is just exactly what I was talking about. The action potential is more likely here, and inhibitory, it's less likely. And they can happen at the same time, and that's called summation when you add them together. And the net effect leads to a greater possibility of action potential. There's some few ways to look at this. So this is what we're talking about. We're at rest, and an excitatory stimuli causes you to go towards negative 55 is threshold. So it got close to threshold. An inhibitory stimulus, an IPSP, is going to move you away from threshold. So the odds of you getting here are much less. There are, they think, about 100 neurotransmitters. We know acetylcholine as one because it stimulates your skeletal muscle to contract. Neurotransmitters can be made of single amines. They can be made of amino acids and peptides. They're usually proteins, and they are produced in the reservoir or the cytoplasm. And when the impulse reaches the synaptic knob of the axon, those neurotransmitters are released by exocytosis. And you don't need to worry about vesicle trafficking. That is cray-cray. So here is a neurotransmitter, acetylcholine, and it controls skeletal muscle actions in both central nervous system and peripheral nervous system. And then there's like other neurotransmitters, and you've heard of these, right? Norepinephrine, dopamine, serotonin, histamine. So all of these also affect central nervous system, peripheral nervous system, or both, and in different ways. You do not need to memorize how these affect the body. There are GABA. That's a big one right here. GABA is a common one. Again, you don't need to know it, but I just want you, nitric oxide, that's huge. This is plays a role in memory, also in vasodilation. This is what makes you have an erection. I mean, like, that's pretty important, right? So I just saw a commercial the other day about taking something to increase your nitric oxide. Helping memory is what they were talking about. But, old folks might need help here too. Sorry if that offended you. There are so many disorders associated with imbalances of neurotransmitters. Depression and epilepsy. Oh, Huntington's disease is so sad. Hypersomnia, insomnia. I don't have enough serotonin. And so my doctor tried to give me something to help with that. And, it made me so sick. So we're going to try something else. Maniac, being a maniac. Am I one of those? Over-talkativeness, irritability. That might be me. Just kidding. Well, maybe not. Parkinson's disease, schizophrenia, and tardive dyskinesia. Drugs that alter neurotransmitters. Ooh, right, tryptophan, remember, that's in your turkey, and so that affects your serotonin. And right after Thanksgiving, what does everybody do? They fall asleep watching the football game. Valium, nicotine and cocaine, other reuptake inhibitors. Drugs affect neurotransmitters, and I'm going to show you a fun thing in a minute. Events leading to neurotransmitters release. Okay, so that's a table for you. Let's go ahead and look at that thing I wanted to show you. So let me pull up. Okay. This is called Mouse Party. Oh, I hope it will work on my Mac. I don't know. So this was made by the University of Missouri, maybe? I don't know that it's like Mouse Party. Try Meth Mouse. Okay, let's try this. Oh, gosh. Oh, yeah, it's working. So these mice have had a party, okay, and we're going to look inside their brains and see what's going on. They've all done a series of different kinds of drugs, and we can see how these drugs affect the body. So which one do you want me to pick? Is this your favorite? Everybody likes the one on ecstasy. So let's just start with him. Or her. I don't know the gender of this mouse.
Serotonin transporters are responsible for removing serotonin molecules from the synaptic cleft after they have done their job. Ecstasy mimics serotonin and is taken up by serotonin transporters. In fact, ecstasy is more readily taken up than serotonin itself. This interaction with ecstasy alters the transporter. The transporter becomes temporarily confused and starts to do its job in reverse. The transporter starts transporting serotonin out of the cell. The excess serotonin becomes trapped in the synaptic cleft. As a result, it binds again and again to the receptors, overstimulating the cell. Ecstasy affects serotonin pathways responsible for mood, sleep, perception, and appetite. Ecstasy also indirectly interacts with the reward pathway. The excess serotonin stimulates a milder release of dopamine along the reward pathway, giving ecstasy slightly addictive properties.
And we won't do all of these. But feel free to, oh, it's Utah. Pick whichever one. Let's look at alcohol. Oh, he dropped his bottle.
Inhibitory neurotransmitters called GABA are active throughout the brain. These neurotransmitters act to control neural activity along many brain pathways. When GABA binds to its receptor, the cell is less likely to fire. Meanwhile, in another area of the brain, another neurotransmitter called glutamate acts as the brain's general-purpose excitatory neurotransmitter. When alcohol enters the brain, it delivers a double sedative punch. First, it interacts with GABA receptors to make them even more inhibitory. Second, it binds to glutamate receptors, preventing the glutamate from exciting the cell. Alcohol particularly affects areas of the brain involved in memory formation, decision-making, and impulse control.
Okay, so feel free to go back and do some partying, not in real life, okay? Mouse partying. So many neurons in the brain and spinal cord make neuropeptides, and they can act as neurotransmitters. Not going to test you on this slide at all. But opiate drugs like morphine, heroin, codeine are all potential painkillers, and they're made from the poppy plant. We have our own opiates called endorphins, and endogenous opiates relieve pain in acupuncture, so they cause you a little bit of pain, and that gives you some opiates. Analgesia during childbirth happens, a feeling of well-being during exercise. You're releasing endorphins all during those activities. So opiate drugs are very useful in getting rid of the pain, and so it is addictive. Let's see. I don't think we need this, so X this out. We're not doing neuronal pulse. But we will do convergence and divergence. Have you ever read that book, Divergent? Okay, well, anyway, convergent is when one neuron receives input from several neurons. So these incoming impulses represent information from different areas, different receptors, and the nervous system can collect, process, and respond to all that information. It makes the neuron able to sum all those impulses. Divergence is one neuron sends impulses to several neurons and it branches out, and this could amplify an impulse. The impulse from that sensory receptor could reach different regions of the central nervous system for processing. So this is convergent, coming together as one, and divergent, branching off into more than one. So drugs, as we learn, do alter the activity of our neurotransmitters, or enhance them from being released, or stop them from being released. Antagonists are drugs that bind to receptors and block the neurotransmitter. Agonists are drugs that activate the receptors. Nicotine binds to nicotine acetylcholine receptors, and the channels for those ions open, and that leads to a dopamine release and pleasurable feeling. And that is all for that chapter. Hope you enjoyed it and hope you learned a lot.